Hello everyone. Today we will discuss about beta-lactam ring synthetic pathway. This beta-lactam, so-called as beta-lactam pathway, and this beta-lactam pathway is particularly found in case of bacteria and also in case of fungi. Okay, so this beta-lactam pathway, uh, beta-lactam lactam, I would say pathway or beta-lactam ring pathway, whatever you can say. So this beta-lactam pathway is meant for the production of a beta-lactam ring compound. Beta-lactam ring compounds. Okay. So what is beta-lactam ring compounds? So these type of compounds have a, you say, a very common structure, that is beta-lactam ring. This beta-lactam ring is a four-ring structure, four ring uses structure, okay, in which you can find at the beta carbon atom, at you say beta carbon atom, there is you say NH group, that's the amine group it you say attached. So here you can see the keto group is here, that at this place particularly the structure is something like this. Okay, again it is you say NH is here. Okay, now here there will be, you say, H, there will be C here. So if you say see the structure, it is something like this, and that's why this is so called as, you say, alpha carbon atom. And in this alpha carbon atom, or you say chiral carbon atom, there is a different type of groups is attached here at four different, you say, positions. That's why this is so called as alpha carbon atom. So adjacent to the alpha carbon atom, there is another functional group it attached as NH. That's why this will be the beta carbon atom. So this NH group which is attached there at the beta carbon atom of this four ring structure, that's why this is so called as, you say, beta lactam ring, or beta lactam ring, or this is the common structure which is found in the beta lactam antibiotics. The simplest form of beta lactam, you say, antibiotic, that is beta lactam, you say, compound, that is 2 azetidione denone. So this is the very simplest compound that is found uh, among all, you say, possible, you say, compounds that are having the beta lactam ring or beta lactam compound. Beta lactam is you say found mostly in actually fungi, but also found in case of bacteria for the defense mechanism actually. So this beta lactam, you say, antibiotics or beta lactam, you say, compounds are very common in, you say, penicillin species, and you are more familiar with the penicillin species because you have studied about, you say, discovery of penicillin or antibiotic, world first antibiotic, that is penicillin, which are discovered by, you say, Alexander Fleming in 1928. So all total, there are four different type of, you say, beta lactam, you say, compounds of beta lactam antibiotics are found. One is, you say, penicillin, another one is, you say, monobactam, and third one, that is cyclosporin, and the fourth one carbapenems. In penicillin, or in all the structure, you can see the three part is there. One part you can see here, this is one part, okay, this is second part, and this is third part. So the center part is actually the beta lactam ring, and adjacent to the, you say, beta lactam ring, there are actually two, you say, ring structures are attached, one on each side of the beta lactam ring. One is, you say, amide ring. This amide ring is having actually, you can see here the keto group C double bond O. Okay, now it is bonded with the NH2 here, NH2 here. Okay, so all total this is presenting the amide group. And another one is sulfur containing, you say, ring, that is so called as thiazidine ring. Okay, so in penicillin this is having three, you say, major, you say, region in its chemical structure. The center region is beta lactam ring, two adjacent region, that is one is, you say, amide plane, and another one is thiazidine ring. So all together they are making one structure, one complex structure, you can say, that is, you say, penicillin antibiotic. Then in this case also monobactam, you are having this you say one structure here starting from this to this, and this one is your second beta-lactam ring, and this is your thiazidine ring. Similarly, you can see here in the cephalosporin, this is one structure, and this one is the beta-lactam ring, second one, and this is thiazidine presenting the you say sulfur here. This one is amide, and this one is beta-lactam. Okay, and similarly in the carbapenem also you can see here this part is your you say amide plant, and this one is your actually beta lactam ring, and this one is actually your thiazidine rings. Okay, so coming to the next, this is a schematic diagram, or you can say flow chart, where you can see here the beta lactam antibiotics. They are classified into four major categories that we have already discussed here, and these are the examples of you say different type of antibiotics which are coming under you say penicillin, or coming under cephalosporin, carbapenem, monovectams, and in specifically you say cephalosporin, there are four generation of you say antibiotics are there depending upon user discoveries. Those antibiotics which are discovered at early stage, so called as you say first generation, and in second generation those are you say discovered just after the first generation of antibiotic, and now it become you say third generation and fourth generation antibiotics are you say discovered, that is coming under you say cephalosporin. This is a very you say wide type of antibiotics that are used in the pharmaceutical industries particularly, and also the penicillin now. And now come to, before going to the pathway, we have to discuss that how this beta lactam pathway or how this ability to you say generate or synthesize the beta lactam type of antibiotics are you say found or are developed inside the you say fungi and inside the bacteria. There is two hypotheses are there. One hypothesis is actually a horizontal gene transfer method, that is HGT, or you can say this is nothing but your transformation method, transduction method, and you say conjugation method or sextuction method. So all these are you say different type of you say genetic recombination method that are found in case of bacteria, or you have studied in the bacterial reproduction part. So horizontal gene transfer can be possible bacteria to use a bacterial cell, or bacterial to use a fungi cell, bacteria to use a plant cell. So the gene can be transferred from different uses cell to of same category, or from this a different category. Okay, or bacteria, or you say virus to bacterial cell also possible, or bacterial to viral cell is also possible. So there are different you say cell communication and cell interaction can be possible, where the cell can be fused together, the cell can transfer its gene to another cell. So this kind of situation may you say happen in natural condition. Okay, that is also can be possible in the artificial condition, the laboratory condition. Okay, so that is how the gene transfer can be possible from bacteria to fungi. Okay, because initially the bacteria in the evolutionary you say process, the bacteria are actually first evolved before the actually fungi. So somehow the bacteria is also having the beta lactam pathway, and in due course of time, there is a chance that the bacteria, this genomic entity or the genes from the bacteria, that is transferred to the you say fungi through this you say horizontal gene transfer method. This is one possible way. Another possible way is actually vertical descendant. That is what is saying actually that when the fungi are evolved, there must be a you say origin point of fungi where a you say ancestral species is there, or you can say the common ancestral species is there, where the fungi and the bacteria both are you say segregated from that point. So let's say this is the ancestral species from where the bacterial divergent and you say fungi also divergent here, and from this fungi different type of you say now divisions are coming out, or we have discovered. So there may be chances that this ancestor is having you say beta lactam you say pathway, or having the genes that is actually you say um, meant for the beta lactam antibiotic production. Now when from this ancestor to different type of, you say, category of species evolved, there is a possibility that these may also having the same type of, you say, ability as that their, you say, ancestor do have. Coming to the pathway of, you say, beta lactam pathway that is happening in the fungus, we have taken an example of here penicillin and cephomycin biosynthesis in ascomycota, particularly the mold species of penicillin, or you say penicillin mold species. So here in, you say, penicillin, the L-lysine will be the precursor molecule from where the pathway will be started, and this L-lysine is produced in case of fungi by a pathway so called as alpha amino adapate pathway, through which actually the L-lysine is produced in case of fungi. But when it is conducted in bacteria, this L-lysine is produced by a DAP pathway so called as diamino uh, pamylate pathway that is happening in the bacteria, and this L-lysine is produced from, you say, aspartate, particularly in case of bacteria. Now this L-lysine is converted to, you say, piperidine 6 carboxylate. This is the compound which is actually developed from actually L-lysine. Now this piperidine L carboxylate is converted into three different compounds. One is alpha amino adapic acid, and the second one L-cysteine and L-valine. All these three are, you say, non proteogenic, you say, amino acids. When you say it is all are, you say, non proteogenic amino acids, that means these are not involved in the, you say, production of proteins. And in this pathway, these three, you say, component, or these three, you say, non-proteogenic amino acid, they serve as the building block for this beta lactam pathway or penicillin pathway. So altogether, uh, these three, you say, components, they will synthesize or they will combine to form, you say, SCV complex. Okay, this SCV complex means that is delta L alpha amino peptidyl L cysteine D valine. Okay, this is SCV, and the enzyme which is involved in the conversion of this SCV, you say, compound, that is SCV synthesis. Simply, as SCV is synthesizing, that's why the enzyme should be the NCV synthase. Now this SCV which is converted to, say, isopenicillin, this is a major step, I can say. Until unless the isopenicillin N is not synthesized, there will be no such conversion of further antibiotic production in this pathway. So here actually the ACV, the compound which after formation it is converted by, you say, IPN cyclase. This IPN cyclase, the full form is actually isopenicillin N cyclase. So as the next compound which is equally synthesizing, that is isopenicillin N, that's why the enzyme here it is so named as, you say, IPN, you say, cyclase. Now you can see here in the isopenicillin N compound, there is also a ring that is, you say, thiazide ring. So this IPN cyclase, it actually aids this thiazide ring by replacing the hydrogen here, you can see, and also this part, the D part here, you can see. So that is how this cycle IPN cyclase that edit those you say sulfur ring to the, you say, isopenicillin you say N, or you can say IPN compound. Now this IPN, or you say isopenicillin N, is directly converted, can be directly converted into penicillin G directly through the enzyme that is acyl transferase. That is the conversion of this compound to penicillin G of the similar structure. Also it can be converted to two different type of compound. One is, you say, cephobacins, another one is, you say, cephomycin. The two compounds which are also generated, or you can say produced from this conversion of this isopenicillin in different, you say, pathways by modifying its chemical structure. And after the formation of, you say, cephobacin and, you say, cephomycin C, both are combined together to form, you say, cephalosporin, which is another category of antibiotic that is also coming under you say beta lactam antibiotic. So the significance, if you go for asking the significance of the beta lactam pathway, both in case of bacteria and you say fungi, in all in both the cases, this is meant for the you say defense mechanism, that it protects the fungi, it also protects the bacteria. So when they are you say growing together, so the bacteria can you say some bacteria which are having this beta lactam pathway, they will generate the antibiotic, and also they can you say grow over the fungi. Some fungi they also because when the bacteria grows upon the some fungi species, like you say ascomycota species, they will you say resist over the bacterial growth by producing this kind of beta lactam antibiotics. So these are the significance of the beta lactam antibiotics. Thank you.